All right, now let's look at some deep cerebral structures, and we'll start with the basal nuclei. And the basal nuclei are shown here in red, and we're going to have three nuclei that comprise these nuclei: a caudate, which means tail, and that is this sort of curved nucleus that arches around, and it has both a head portion and a tail. So we have the caudate of the caudate, so to speak, the tail of the tail. And then inside of that, we're going to see two nuclei that together will form what's called the lentiform nuclei. And lentiform means lens-shaped. And we'll look at those in just a second in coronal view. But notice the relationship of the basal nuclei to the thalamus. So they really sit just outside of it. So if we were to look at our brain model, and we take up sort of the inner part here, we remember our thalamus here, and we've got the thalamus shown here as well in orange. We then have the basal nuclei here. So this would be the caudate nucleus right here. And we'll see in just a little bit what it looks like in coronal view. So here's our coronal view. And here we have our caudate nucleus that curves around. Here are our lentiform nuclei that are consisting of the putamen and the globus pallidus, so named because it is a pale, round structure. So it looks sort of like a pale globe in coronal section. Now we also have some white matter tracts that are some good landmarks here. Here we have what's called the corpus callosum, which we've been over before. It's one of our white matter commissures that are linking hemispheres of the brain, the two hemispheres of the cerebrum. Then we have something called the corona radiata, which is basically just white matter tracts that are traveling through. They're going to travel through the basal nuclei, through this little highway, so to speak, called the internal capsule. So the corona radiata is just going to be white matter tracts that are going to be connecting the cerebral cortex with lower structures, and we're going to have those white matter tracts be able to run some way through the basal nuclei to get to the lower structure, so they run through this internal capsule. So all of this is the internal capsule. We'll also notice this other structure called the amygdaloid body, and we can see it here as well, sort of at the anterior portion at the tail of the caudate. And it is typically grouped with limbic structures. And this is going to be very important for assessing fear and threat situations. So it's going to assess the threats, and it's going to modulate your reaction to them. So it's a very important nucleus, and when it's damaged, sometimes people will start to show aberrant behaviors to noxious or threat-causing stimuli. Now let's look a little bit again at this brain model, because this is the caudate. This here would be some representation of some of our white matter fibers that will be running from the corona radiata through this internal capsule here. Now the function of the basal nuclei is subconscious adjustment of ongoing motor movements. So it's basically going to put the brakes on, so to speak, some of the commands issued by the motor cortices, and it's going to adjust the force and velocity of these movements so that they're not just some herky-jerky movement out there and we don't have uncontrolled muscle movements. So a lot of times people who have damage to these areas will not be able to coordinate or control their muscle function very well. And they're also very important for initiating movements. And we will see that the basal nuclei are connected, as you recall, with the substantia nigra from the midbrain. So some researchers group the substantia nigra right along with the basal nuclei because they're working together to adjust the motor tone of these ongoing motor movements, these conscious motor movements, but the basal nuclei are constantly adjusting and fine-tuning these movements. Here, once again, is your amygdala, and there again, that's important for threat assessment and basically modulating fear responses. Now let's look at some other structures called the limbic system, which we've already touched on, and the amygdala, which is shown right here, is actually considered part of the limbic system. So we're going to have some cortical structures that you can really only see in sagittal section. So we're going to have the cingulate gyrus, so called because cingulate means belt, and the cingulum runs right over top of the corpus callosum here. We also have something called the parahippocampal gyrus, which is on the interior surface of the temporal lobe. So that would be on the interior surface, which we really can't see on our brain model, but we can see it here. And the parahippocampal gyrus is going to contain this red structure called the dentate gyrus. And the dentate gyrus contains a structure called the hippocampus. And the hippocampus is so called because it looks like a seahorse. Or at least anatomists thought it looked like a seahorse, so they gave it the Latin name for seahorse, which is hippocampus. Now, the hippocampus is very important for two major functions, one of which is the consolidation of short-term memory into long-term memory. So that is, if you're trying to take a fact that you've just learned and turn it into something that you'll remember 2 weeks from now, 2 months from now, 2 years from now, then you're going to have to consolidate that from short-term memory to long-term memory. So when you think of short-term memory, you think of things like memorizing a telephone number long enough to dial it, and then once you reach the connection, you forget it. You don't need it again. But other things, other bits of information, like what you're studying through your anatomy and physiology course, you want to remember it for a long duration. So hopefully you'll be able to remember it for the rest of your life. Ideally, you would hope so. So in order to get that information out of your short-term memory and put it into long-term memory, which is a process called consolidation, we need the hippocampus in order to do that. Without the hippocampus, you can't accomplish that task. And there was a very famous patient who went by his initials HM, and he had a bilateral hippocampectomy. So in other words, they took it out completely. He had a situation where before the hippocampectomy, he could remember all the events of his life up to that point. After that point, he couldn't remember anything new. He couldn't learn any new facts, and he couldn't lay down any new episodic memories about his own life. So he could have a conversation with a doctor who might come in and interview him for some time. She could introduce herself, and they could play games together, and they could do all kinds of things and spend several hours together. Then she could leave. But because he couldn't put any information from short-term memory into long-term memory, she could come back in the room 30 seconds later, and he would have no idea who she was. However, his procedural memory, that is, being able to do physical tasks, was unaffected. He could still learn to do new things, physical things, that didn't require any fact or episodic memory, but he couldn't lay down any new ideas or information about the episodes in his own life or new facts that he learned. The hippocampus is going to be strongly interconnected with regions of the hypothalamus. So we have a white matter tract here called the fornix, which links structures of the limbic system with structures of the hypothalamus. And one of the structures that are strongly connected are the hippocampus to the mammillary bodies. And while we already have learned of the mammillary body's role in mediating reflex eating movements, they also have a role in consolidation of learning and memory and work with the hippocampus. We also have another function of the hippocampus that's not really talked about in the book, and that's spatial navigation. So both in humans and in other animals, if we're able to record activity from cells within the hippocampus, we will find that if you put animals, say a rat in a maze, and you put electrodes all along the hippocampus and record from individual neurons, what researchers have found is those individual neurons will start to become active when the animal enters a certain area of the maze. So the certain areas, certain cells within the hippocampus will come to represent certain areas within the maze. And there have been quite a bunch of interesting research studies done both on animals and on humans examining the role of the hippocampus in spatial navigation. So the limbic system has a lot more function than just mediating emotional states, as we've thought of it before and we've talked of it before, but it also is going to be important for consolidation of short-term memory to long-term memory and also spatial navigation. What's interesting about the hippocampus is that whenever you have a very strong emotional reaction to something, you're much more likely to remember it. So the hippocampus is going to be much more active laying down bits of information if there's a strong emotional valence that's tied with it. Another interesting fact is that we're going to have some of our olfactory cortex very close alongside parts of the limbic system and the hippocampus. And so smell is another really potent thing that can evoke memories. So smells can evoke very, very strong memories.